Hi everybody. Today I'm going to be talking about cellular respiration. So cellular respiration occurs sort of around the mitochondria. The mitochondria has two lipid bilayers. It creates this internal region, which is called the matrix, and this region here called the intermembrane space. So cellular respiration begins with breaking down sugar in a process called glycolysis. And glycolysis actually occurs outside of the mitochondria. Into glycolysis, we have to put in a glucose molecule. We have to invest energy in the form of two ATP molecules. And we need two electron carriers called NAD+. And out of glycolysis, we break glucose into two molecules of pyruvate. We gain 4 new ATP, and our two electron acceptors get converted into two NADH molecules. This is the reduced form, and glucose is oxidized into pyruvate. So after this, the pyruvate will move into the mitochondrial matrix for pyruvate oxidation. So into it, we put pyruvate, and we put an NAD+, and we put a molecule called coenzyme A. Later I will abbreviate this as CoA. So we put this in, and out we get acetyl-CoA, which is just 2 carbons attached to this CoA group. We get the reduced form NADH, and we get a carbon dioxide. So after pyruvate oxidation, we send acetyl-CoA into the Krebs cycle. This is also called the citric acid cycle, and it's also sometimes called the tricarboxylic acid cycle. So they all mean the same thing. Krebs cycle is kind of like a little circle, just keeps going and going. It has many steps, but overall the main idea is that we put in acetyl-CoA, we put in 3 NAD+, and we put in 1 FAD, which is just another kind of electron carrier. So out of this, we get 2 carbon dioxides, we get 3 NADHs, we get 1 FADH2, and we get our coenzyme A back, which can go back and do this into the pyruvate oxidation. Okay, so now we have completely broken down our sugar. Note that there's 2 pyruvates made per glucose molecule, so pyruvate oxidation can happen twice per glucose molecule, and the Krebs cycle can also happen twice per glucose. So after we've gone through the Krebs cycle and all these other steps, we have all of these electron carriers that are now reduced. But we want to use them again. We want to keep reusing them. So we go through what's called the oxidative phosphorylation. Oxidative phosphorylation has two parts. It has the electron transport chain, and it has chemiosmosis. So I'll start with the electron transport chain. So over here in the intermembrane space, there are a couple of complexes, 4 complexes of proteins. And your electron carriers will go into the electron transport chain. So I'll put in all of our NADH and FADH2 molecules. And the electrons will get dropped off into one protein complex and go on through increasing reductive potential. While the electrons are being dropped off, it's also putting protons into this intermembrane space. I'm going to draw them all over. So the intermembrane space, because it's in between 2 lipid bilayers, these protons can't get out, so it creates a proton concentration gradient inside the IMS. So we go through our electron transport chain, and at the last stop, electrons need to go somewhere. So this is oxygen's job. The electrons end up on oxygen. Now, oxygen with tons of electrons on it isn't very happy. It's unstable and reactive. So we will take a couple of protons that were floating in the matrix, and we'll get to why in a second. But we take protons from the matrix and put them on this oxygen to ultimately create water. So what about these protons in the membrane space? I'm going to move this over here just for space. Have some protons. Now, we talked about how glycolysis, cellular respiration is to make energy. So most of the energy doesn't come from glycolysis. Oh, I forgot that the Krebs cycle also makes 1 ATP. So we have an overall of 2 ATPs from glycolysis and 1 from the Krebs cycle, 2 for glucose molecule, because it goes through twice. But that's not most of the energy. That's not very much. Most of the energy comes from this right here. This is a protein called ATP synthase. So these protons, they really don't like this concentration gradient. They really want to be in the matrix. But the only way for them to go through is to go through this protein complex. And this protein complex powers a little motor that converts ADP and an inorganic phosphate to make ATP. So just to run through that one more time really quickly, glycolysis occurs in the cytoplasm. We take 1 glucose molecule, we invest 2 ATP, and we use 2 electron carriers, NAD+, to break glucose into 2 pyruvate. We gain 4 ATP, and we produce 2 NADH. The overall gain of ATP here is 2. We take it into pyruvate oxidation. So our pyruvate that we made goes in. We use NAD+ and coenzyme A to create acetyl-CoA, NADH, and carbon dioxide. The acetyl-CoA then goes into the Krebs cycle, along with 3 NAD+ and 1 FAD. And out of this, we get 2 carbon dioxides, 3 NADHs, 1 FADH2. We get our coenzyme A back, and we get 1 ATP. All of these electron carriers that we have produced need to be reused and recycled. So we will drop off all of the electrons into the electron transport chain. As they go through the electron transport chain, they will pump protons into the intermembrane space. And at the very end, they will deposit the electrons onto oxygen, which will then grab protons and become water. These protons in the intermembrane space will go through ATP synthase, which will power a proton pump, or it is a proton pump, and it will power the spinning of a motor to produce ATP. So we discussed what happens through cellular respiration when oxygen is present, because oxygen has to be the terminal electron acceptor. But what if there's no oxygen? That happens when you work out really hard in your muscles. It also happens for lots of bacteria who can't use oxygen. So there you use a process called fermentation. So you start off with glycolysis again. It's the same glycolysis. So you're still putting in your glucose and your 2 ATP and your 2 NAD pluses, and you still get out 2 pyruvate and 4 ATP and 2 NADHs. But if we want to continue with glycolysis, we need to recycle this. But there's no electron transport chain because there's no oxygen. So what we'll do is we'll go through fermentation. There are 2 kinds of fermentation, but they both undergo essentially the same type of thing, so I will discuss them together. So you put in your pyruvate, and you put in your NADH. And out, you will always get NAD+, which can then go back into glycolysis. And you can get one of 2 products, depending on what kind of organism you are. If you're a human and you worked out really hard and your muscles are sore, you will create lactic acid. Or if you're a certain kind of bacteria or yeast, you will create ethanol and carbon dioxide. So we've gone through oxidative and non-oxidative. So thank you very much.